So this morning we're going to kind of switch topics. We've been talking about surface area to volume ratio and movement of gases and nutrients and waste in and out and around the body. Now we're going to talk about how marine fish regulate their ions in their body and regulate the movement of water in and out of their body based on those ions. So before we get any further than this, we need to have a quick review of osmosis. So different bodies of water have different salinities and it can change constantly. So the organisms need to be prepared to be in different bodies of water. And this is because of the concept of osmosis. So as a review, osmosis is the movement of water from high to low water potential. So this is similar in concept to a concentration gradient, but you guys need to understand this new vocabulary term called water potential. So water potential is just the potential energy of water in a solution, and it comes into play. So if you look at this diagram over here where I'm highlighting is the area of high water potential. So there are more water molecules than there are sugar molecules in this case. We would think about it like salt, but it's the same concept. So on this side of the semi-permeable membrane, there is more water molecules. So this would be considered a high water potential because there are more water molecules here than salt molecules. On the right side of the semi-permeable membrane is an example of low water potential. So the red dots again are our water molecules. There are less water molecules over here. So what would happen, water would be allowed to move from the left side into the right side here. Essentially, the water wants to dilute the salt that's over here. So the osmosis direction is going to be moving from the left side to the right side in this particular case. So how does this apply to what we're about to talk about? It's important to remember that cells actually contain dissolved salts. We have sodium chloride and potassium ions are really important ions that are contained in both the cells and the interstitial fluid in our bodies and in the bodies of marine fish. So when these cells are submerged into a liquid, osmosis becomes a player in the game. So if you look over here, we're focused on the animal cell model up here. So hypotonic solution. Hypo means less, so there is less salt in this outside water here. So this would be an example of like fresh water. So in a freshwater environment without the ability of regulation of the ion concentrations in the cell, this would happen. Too much water would continuously flow into the cell because this is our high water potential outside, low water potential inside, and the water would eventually burst the cell. An isotonic solution is a solution where the salt content is the same on the outside and the inside of the cell. So this is, we're going to see that a lot of marine invertebrates actually just take on whatever salinity is outside of the cell, and so they are able to have an isotonic solution, and that's how they are able to deal with it. And then a hypertonic solution would be a super salt solution on the outside, meaning that the high water potential inside the cell would have the water move from inside the cell to outside the cell, and therefore the cell would shrivel up. Okay, so this movement of water is why marine species are sensitive to changes in salinity of the water. So again, if you take a look at this table, we've got several different types of organisms here. You'll notice the invertebrates, mussels, and jellyfish has body fluid as their example because they don't have circulatory systems. The toadfish, the eel, and the salmon are bony fish, and you'll see that's plasma. Plasmid is the liquid portion of the blood, so it's the fluid portion of the blood. So if you look here, we've got seawater, the approximate, let's focus on sodium, the approximate sodium concentration is 450. If you look, mussels, jellyfish, and seawater all have about the same. So body fluid of invertebrates is typically similar to that of the seawater, and by being an osmoconformer like this, they don't try to regulate the salt concentrations, they just go with it. So they end up, their body fluid ends up being isotonic to the surrounding fluid. If you notice, the bony fish have concentrations that are significantly less than that of the seawater. So without any kind of regulation, what would happen is the water would leave the cells of their body and move out into the water, which is actually what happens, but they just have mechanisms built in to regulate or to make up for the fact that they're losing all this water like this. So again, to talk about the invertebrates, they are osmoconformers, meaning that they conform to the osmotic pressure of their outside environment, or that they stay that they become the same as the environment that they're in. So the water enters and leaves their bodies equally, so there's no overall change in their water content. The bony fish, however, are going to have the osmotic concentrations that are different than that of the seawater. So they have to have some sort of mechanism in order to deal with this changing, this water leaving their bodies. So this brings us to our next one, which is how do marine bony fish osmoregulate? And we're going to limit this to drinking seawater, absorbing water through the skin, and then actively excreting salt. They usually do that through their gills and through urine, and then also using energy from respiration to actively pump some of these ions out of their body. So again, osmoregulation is when organisms maintain the solute and water content in their bodies. Marine bony fish are, in most seas and oceans, water surrounding the fish has a higher salinity than the cells inside of the body fluids. So when water is drawn out of the body from the gills and skin by osmosis, and then the salt diffuses into the body from the water. So these fish are dealing with constantly losing water from their bodies, which could lead to dehydration. The way that they get around this is to constantly drink seawater, which replaces the water that's lost by osmosis. So they actually drink the seawater. The sodium and chloride ions are actively pumped out of their bodies through their gills. So again, that transport system is going to be important for bringing those ions up towards the gills. And then there are actually pumps, active transport pumps, which means they use ATP in order to pump salt against the concentration gradient. So it costs them energy, but it pays off in the long run. They also have the ability to excrete some of these ions through the urine. So the kidney takes and concentrates these ions in a small volume of water, because remember, they don't want to be having diluted urine, because diluted urine, it means that they're losing more water. So they have highly concentrated urine that has a lot of these salts in them, so they're getting rid of more salts that way. And then the kidney actually reabsorbs the water that they are, when the kidneys are processing the salts, the kidney kind of hangs on to that extra water, so it continues to hydrate them, and they get rid of that concentrated salt solution. All right, so here's a quick breakdown. We've got this fish over here, and we're going to look at a better diagram on the next slide. However, you can see this is for marine fish only, but marine fish drink the seawater, active pumping of the sodium and chloride ions out of the gills. They do lose water out of their bodies by osmosis, but again, they're trying to make up for that loss there by drinking the seawater. And then they have water, small amounts of water, and then highly concentrated with the salts in their urine. So that's how they're trying to get rid of that. Again, you can see here we've got the tuna. It's the same concept. This gives a little bit more detail. I like this one better. So if you would like to take down any of these notes, go ahead.